Welcome to Biology at Priyojit, my dear viewers. Today we will learn about 3' polyethylene of eukaryotic mRNA, which is an important post-transcriptional modification in which a long chain of adenine nucleotides are added to the 3' end of each mRNA. This polyethyl increases the stability of mRNA, helps in its transport from the nucleus to the cytoplasm, regulates its lifespan, and plays a crucial role in the initiation of protein synthesis. Introduction. Polyadenylation is a post-transcriptional modification that occurs only in the eukaryotic mRNA except histone mRNA. In this process, a long chain of adenine nucleotides is added to the 3' end of the mRNA, forming the polyethyl. What is the importance of this polyethyl? It is essential because it increases the stability of mRNA, helps in termination of transcription, facilitates initiation of translation, it regulates mRNA turnover. So half-life, that is time required to degrade 50% of the mRNA, and that is also regulated by the length of the polyethyl. So what are the enzyme involved for this process? First one is polymerase, which cleaves the newly synthesized mRNA at the 3' end. It adds about 200 adenine or A residues to form the polyethyl, works without a template, that is DNA independent RNA polymerase, and uses ATP as the source of adenine nucleotides. Signal for polyadenylation: when the RNA polymerase 2 reaches the end of a gene, it transcribes a special polyadenylation signal sequence in the mRNA. This signal attracts the polyadenylation machinery from the CTD or C-terminal domain of RNA polymerase 2. What are the major steps of polyadenylation? Cleavage of the RNA at the 3' end, and addition of adenine residues to form the polyethyl, termination of transcription by RNA polymerase 2. So other proteins factors involved in the process: two important protein complexes travel with RNA polymerase 2. Number one is CPSF, cleavage and polyadenylation specificity factor. And number two is CSTF, cleavage stimulating factor. Their roles include: CSTF cuts the RNA at the 3' end and then dissociates. CPSF remains bound to the RNA and recruits PAP or poly-A polymerase. Poly-A polymerase also adds the polyethyl. So finally, poly-A binding protein binds to the polyethyl and removes poly-A polymerase and CPSF, that is cleavage and polyadenylation specificity factor, and protects the mRNA from degradation. So this is all about polyethylene. So as we conclude, that is 3' polyethylene is a vital modification of eukaryotic mRNA that enhances its stability, aids in nuclear export, and promotes efficient initiation of translation. By regulating the lifespan and usability of mRNA, the polyethyl plays a key role in controlling gene expression. This simple addition of adenine residues has a profound impact on the proper flow of genetic information. So keep exploring the wonders of molecular biology, my dear students. Thank you.